importance in differentiating different types of meningitis. For that, you should know the composition of CSF and how the composition gets altered in different types of meningitis and brain tumors. Let's uncover all about CSF video. Now, CSF refers to a clear colorless fluid that is synthesized by choroid plexus, and it surrounds the brain and the spinal cord. So, the circulation of CSF is as follows. It is synthesized in the choroid plexus and gets released into lateral ventricles. From there, it reaches third ventricle, fourth ventricle, and gets distributed across the subarachnoid space covering the brain and the spinal cord. Now, why do we need CSF? This acts as a shock absorber, it acts as a nutrient supporter, removes the waste products from the circulation, and also maintains the intracranial pressure. Now, coming to the composition of CSF, it is appearance is it is clear and colorless. Pressure is around 70 to 180 water. Protein concentration is 10 to 30 milligram per deciliter, very less when compared to the plasma. Check out for the chloride level, it is around 112 to 132 milligrams per liter. The chloride concentration of CSF is more than that of the plasma. This is because CSF protein is very less. In order to maintain the Gibbs-Donan membrane equilibrium, chloride, which is anion, its concentration is increased. Now check out glucose is around 50 to 70 milligram per deciliter. This is because now glucose enters the brain and the spinal cord through GLUT1 transport protein. Now this glucose is continuously required by the brain for its metabolic needs, so the levels are always very less when compared to the plasma. Coming to the cells, there are only 0 to 5 into 10 power 6 per liter of the different types of cells are seen. Cerebrospinal fluid. Now, the CSF analysis is of great importance to differentiate different types of meningitis. Now, this cerebrospinal fluid is obtained by passing a sterile needle between L2 and L3 or L3 and L4 lumbar vertebra, and this fluid is subjected to biochemical analysis and also microbiological analysis. So, basic biochemical analysis to differentiate different types of meningitis are as follows. In bacterial meningitis, the appearance becomes turbid. CSF appearance is turbid. Cells are increased, proteins are increased, sugar is decreased because bacteria use up the sugar for its metabolic need. And when kept for a long time, clot is formed. That is the coagulation profile in bacterial meningitis. Now, next one is most common in India is the tubercular meningitis caused by Mycobacterium tuberculosis. So, the appearance also is turbid here. There is increased cell count in the form of lymphocytes. Proteins are also increased here, and sugar is decreased because the tubercle bacilli utilize sugar for its metabolic need. And typical coagulation profile is cobweb type of coagulation seen in TB meningitis. This is one of the Viva question asked and also MCQ question that will be asked. Coming to viral meningitis, the appearance is clear here. There is increased cell count, increased protein, sugar is normal, and there is no change in the coagulation. This is how we can just differentiate the bacterial from the viral meningitis, where the appearance is clear and there is no change in sugar concentration in viral meningitis. Now, coming to brain tumor, any tumor within the brain, the color of the CSF remains clear. Cell count is normal, whereas proteins are increased and sugar is decreased, and it will solidify when kept for a longer time. The CSF solidifies. Now, so this the characteristic feature, that is albuminocytological dissociation. It means that there is increased concentration of albumin, and whereas the cells are normal. This albuminocytological dissociation is characteristic feature of brain tumor. So this will definitely help us to differentiate meningitis from brain tumor when we do this kind of CSF analysis. The last part is subarachnoid hemorrhage, that is bleeding into the subarachnoid space. So the what happens, CSF becomes blood tinged or blood stained CSF is seen here. Among the cells, both the normally when compared to WBC, RBCs are also increased in subarachnoid hemorrhage. Proteins are increased, sugar is normal, and there is no change in coagulation. So this simple table will give clue to identify different types of meningitis, tumor and subarachnoid hemorrhage based on CSF analysis of cells, protein and sugar, and also the coming to one more important topic that is related to CSF part is multiple sclerosis. So in multiple sclerosis, what happens is body's immune system attack the myelin sheath. Now this attack of myelin sheath is not continuous and it is discontinuous. The attack is seen in different areas of the brain and the spinal cord. That is the reason why it is called multiple. And sclerosis means there is plaque formation seen in the myelin sheath. That's the reason why it is called as multiple sclerosis. Now, what are the biochemical changes that takes place in multiple sclerosis? So the basic problem is myelin sheath demyelination. So what happens is nerve signal travel very slowly. Whenever it is demyelinated, the signals nerve impulse transmission is very slow, or sometimes it may be blocked if the sheath is completely lost. Now what happens is this results in on and off of neurological symptoms. So the symptoms are not continuously. So whenever there is degeneration, the symptoms appear, and whenever the sheath is getting formed mildly, when it is formed or mild demyelination is there, so symptoms disappear. So now these symptoms depends on the affected nerves, and symptoms can manifest in the form of muscle weakness if that particular neuromuscular transmission is affected. If it involves the area involving the eyes, in the cerebral cortex, vision problems will be there. Difficulty in walking can be there. Cognitive difficulties like alteration in memory, decreased attention span can be there. Bladder and bowel problems are can also be manifested, can also be seen depending upon the type of nerves involved. Now, how to diagnose multiple sclerosis? The standard one is MRI. So, where you get to see multiple demyelinating plaques in brain and spinal cord. So, that's the reason why it is called as multiple sclerosis. Multiple means multiple areas of brain and spinal cord affected, and sclerosis means plaques are seen where the myelin sheath is damaged. Apart from that, we can do CSF, whatever the CSF, apart from identifying the cells, protein and sugar. So, we can also do electrophoretic separation of the CSF proteins. So when it is separated, this is how the pattern we get. So this is the normal pattern. So what happens, we have the gamma globulin region, then beta, alpha, and fastest moving band is albumin and prealbumin. So what happens in multiple sclerosis, there is increased gamma globulin region because of increased intrathecal production of immunoglobulin G, and this presence has oligoclonal band in the gamma globulin region. So this is one of the characteristic diagnostic feature of multiple sclerosis. So apart from that, we have one more index for diagnosing multiple sclerosis which is increased, that is IgG index. Now this takes into consideration CSF, immunoglobulin G and serum albumin divided by serum immunoglobulin G and CSF albumin. So this ratio is indexed in multiple sclerosis. So this was all about CSF analysis and clinical significance of differentiating different types of meningitis brain tumor using CSF analysis. Stay tuned for more such updates. Happy learning. If you like the video, please do like, share and subscribe.